So hello and welcome back everyone. My name is Boris Bernhardt and it's my pleasure to introduce the lightning talks as the last part of the scientific program of this amazing workshop. So the first speaker is Sherri Lee-Jones and I'm looking forward to her talk.
Okay, thank you. So I'll give a quick overview of our 3D atlas of the human hypothalamus on the big brain. So the hypothalamus lies ventrally at the midline of the brain surrounding the third ventricle. It's involved in homeostasis as well as emotional regulation. And it's a very small structure, about 1 millimeter cubed, but it's comprised of a centimeter cubed, I'm sorry, but it's comprised of a number of nuclei that all have distinct functions, many of which are actually sexually dimorphic in both structure and function. So we did a hypothalamus segment, or the hypothalamus segmentation was performed on BigBrain, the 2015 release, which, as you know, is a post-mortem brain of a 65-year-old man that was cell body stained, digitized, and reconstructed in 3D. So we used the annotation and visualization software called Atelier 3D. For each structure that we segmented, the annotations were performed on voxels at 20 micron isotropic resolution, and the segmentations were guided by the May Atlas and the original histological publications. So the segmentations were done manually every 2 to 20 sections, depending on the size and the shape of the structure. And then the tools within A3D allowed us to automatically extract the annotations for unsegmented intermediate sections, which were then manually edited. And then this process was repeated until each section was segmented for each structure. The final surfaces were then smoothed for the creation of the 3D atlas that I'll show you a bit later. So here's an example of how the segmentations were done. This is a zoomed in coronal view of the, at the level of the paraventricular nucleus. So on the left is the big brain, and you can see the contrast here in grayscale between the different subregions like the magnocellular, so the PAMC, the parvocellular region, the dorsal region, and the posterior regions. And on the right, we have the histological publication by Kucherov that demarcates the different regions based on different immunoreactive markers. So in A3D, there's a color map feature, and that really helped with the visualization to see the different substructures of the PVN, and you can see the resulting segmentations that are drawn in different colors here on the left. So this is the atlas. It will be released open source. The nuclei that were segmented include the preoptic nuclei, the paraventricular nuclei, the supraoptic nucleus, the dorsal medial and ventral medial nuclei. We also segmented the mammillary bodies and the major surrounding white matter tracts, so the optic tract, the anterior commissure, and the fornix. So the next step is to map this to ICBM space. We then plan to use it as a template for the segmentation of in vivo MRI scans, as well as a tool to validate an in vivo segmentation protocol of the hypothalamus. So if you're registered for the OHBM conference, you can check out my poster number 1082, and we can interact live on chat if you have any questions. And I'll be on tonight for the poster session that starts at 8 p.m. Eastern time.
Okay, today I will give an update on our MSM surface registration pipeline to bridge atlases across the big brain and the FreeSurfer and Human Connectome project worlds. This is a collaboration between Alan Evans' lab here at McGill and also David Van Essen's HCV group at WashU in St. Louis. Put most simply, our question is, where exactly does a given region on the big brain surface correspond to other well-known reference average surfaces, such as FreeSurfer's FS average and also HCP's FSLR. The big brain surface is histologically derived and is high resolution, extracted at 200 microns, while the other reference surfaces are MRI-derived group averages and are much lower resolution. Several registration tools exist, but each has only been validated or parameterized within its own pipeline. The most recent and flexible of these is MSM, multimodal surface matching. For big brain, we find that standard registration tools commonly lead to catastrophic anterior cingulate and midline misregistrations. For example, you can see my pointer here with standard MSM, the output, the callosal sulcus here in purple, it should be about where these white markers are, but it's been incorrectly dragged back posteriorly onto that flat medial cut. Our solution is to optimize pre-processing at 2 points. First, at inflation of the white surface to sphere, and that affine rotation of the sphere, which is used to initialize the rest of the MSM pipeline. We retessellate the big brain white surface with open flipper, and this allows it to inflate the sphere more flexibly. And then we effectively borrow its inflated sphere, that output, make it the input sphere for the rest of the pipeline. In addition to sulcal depth, we also use the YZ coordinates along the medial cut as a kind of anchor of the problematic regions during the affine rotation of the sphere. The rest of MSM downstream is relatively standard. It's a course-defined approach, starting with a sulcal depth stage. This emphasizes the global features, and then there's a curvature stage, which emphasizes the finer details. Following registration, we can inversely project atlases from FSLR or FS average back to big brain. For example, here I'm showing the HCP MMP atlas from Glasser and colleagues 2016, projected from FSLR back to big brain. We can do the same thing for brainatome, which is defined in FS average back to big brain. And also von Economo, again, back to big brain from FS average. Our landmark analysis shows the accuracy benefit here in green is greater than the accuracy cost in red. So another way of saying that is that by using our modified pipeline, as opposed to the standard MSM pipeline, improvements in regions that were badly misaligned are greater in extent than any small accuracy sacrifices made in other regions. Finally, our mean distortion values are shown here in shades of blue. They're slightly higher than those that have been previously reported for standard MSM on the HCP dataset, but they are much lower than FreeSurfer standard registration tool, which is shown in red. And our maximum distortion values are comparably as low as those previously reported, again, the standard MSM on the HCP dataset. And all of these are much lower than FreeSurfer, again, in red. So in summary, big brain can now serve as an unprecedented cross-validation tool. Macroscopic parcellation boundaries derived from in vivo imaging can be directly compared to cytoarchitectural properties. And conversely, big brain's histological landmarks or cortical layers, such as those shown by Conrad in his earlier talk today, may be transposed back to FSLR and FS average. Of course, we continue to optimize this approach. You can find me at my poster here virtually. Thank you.
Thank you, Lindsay. So next speaker is Jessica Royer, also from the MNI.
So hi everyone, I'm happy to present today our work on in vivo microstructural gradients in the human insula. So in this study, we bridged the worlds of in vivo microstructural imaging and cytoarchitectonics and examined the correspondence of functional connectivity and microstructure within the insular cortex. So we know that the insula is a highly complex region, both in terms of its functional organization and microstructure. So previous studies profiling the functional heterogeneity of the insula have consistently highlighted most prominent variations along its anterior-posterior axis, as well as between its dorsal and ventral anterior subregions. And although investigations performed in postmortem specimens have highlighted a similar axis of change regarding insular microstructure, the direct link between the insula's functional organization and its microstructural composition remains to be established in living humans. So we address this by leveraging myelin-sensitive imaging to derive microstructural gradients along the insular cortex. So to do this, image intensities were sampled across cortical depths and then cross-correlated across all insular vertices. And we then applied a nonlinear dimensionality reduction approach to derive gradients of microstructural similarity across the insula. So this revealed two gradients, a first gradient evolving along the anterior posterior direction and a second gradient in the ventral dorsal direction, which we can then use to form this two-dimensional representation that can be leveraged to track other structural or functional features varying along the insular cortex. So we then harnessed the 100 micron big brain dataset to validate these in vivo gradients against histological measurements. So we found that the anterior posterior gradient followed changes in cytoarchitecture, with strongest correlations found in these deeper surfaces closer to the white matter boundary, and that peaked at 87% depth. And we can observe that histological intensities at this depth were highest in the ventral anterior subregion of the insula, suggesting that higher cell density and soma size in deeper layers of this primarily agranular region of the insula. So we also examined microstructure function correspondence by grounding different functional features within the microstructural gradient space. And focusing on one aspect of functional organization we explored in our study, we found that regional changes in functional connectivity to established network communities followed insular microstructure with increasing affiliation to attentional and executive network communities in the anterior insular cortex. So just wrapping up, this multi-scale examination of the human insula first used myelin-sensitive imaging to derive main axes of microstructural variation in this region. And I should note that these gradients were reproducible in a distinct data set, in a different sample using a different myelin-sensitive proxy. And variations in cell density and soma size reflected in the histological measurements provided by BigBrain allowed us to provide these direct cytoarchitectonic validations for our principal anterior posterior gradient. And finally, we showed that this microstructurally guarded representation of the insular cortex can recapitulate local variations in functional topographies. So if you're interested in the full story, you can check out our paper that's available in the NeuroImage special issue on gradients. And I'll just take a quick second to thank everyone who made this work possible, with a special thanks to my supervisor, Boris Bernhardt, and my dear colleague, Paquola. Thank you.
Yes, thank you very much. Next speaker is Roxana Kooijmans from the Netherlands Institute of Neuroscience and Jülich.
Hi, my name is Roxana Kooijmans, and unlike the vast majority of the speakers today, I'm actually a histologist. So I don't work on the big brain data, but together with Markus, I actually want to present some data that we think could be added to the data that most people are working on, and that could bring a lot of new dimensions to it. So I'm going to start off, if my computer collaborates, with one of the most popular borders, highly featured today as well, between area 17 and 18. So this is what it looks like in a generalized cell body stain, in a Nissl stain. And then this is what it looks like, sorry? This is what it looks like in my preparation. Which is a cell-specific preparation here for 3 different cell populations. So this is possible due to a protocol that I have developed. Here we have an example showing staining for calcium-binding proteins, parvalbumin, calbinin, and creatinine. So we can identify these 3 populations in the same section with different colors, blue, magenta, and brown in this example, and hopefully now it's going to work. So what you actually saw was a high resolution zoom in of a section that is actually a scan of a full human hemisphere section. So now as we zoom out, you can see that this is actually very large. It has very uniform quality. You can see a lot of different areas, and we see this prominent area 17 with this very typical sublayering here with a very prominent layer 4C parvalbumin positive population. And if everything works out, we're going to end up with our favorite border between area 17 and area 18. So building on this, I have also been working together with Erik Opschulte from Timo Dikscheid's group in Jülich, and we have been implementing machine learning in order to generate a kind of quantification that is similar to the gray level index, but it is cell type specific. We can talk about that later. You can contact me. So to further develop this, I'm also working together with Markus Axer. This is a test done in a vervet monkey. So this is a full vervet section in the sagittal plane. This is the PLI that Markus works on. So this shows you fiber and connectivity in the whole brain. And this is the identical same section then stained for different cell populations. So here we fly into the brain again, and we're going to go to the LGN and the pulvinar, but this is just to show that we can now, in the same tissue, actually obtain images in very high resolution, 1.3 microns isotropic in Markus's case, and 1 micron in my case, in the same section, both for fibers and different cell types. That was it. And these are the people that are currently involved in the project. Thank you.
Terrific, many thanks. So the next speaker is Jorge Bosch-Bayard.
My presentation is very fast, and I only want to acknowledge a group, the big group that is working on having electrophysiology on BigBrain. It's a group in which we are participating people from the Cuban Neuroscience Center, from the Montreal Neurological Institute, and from the University of Electronics and Science and Technology of China, where Pedro Valdez has his group. This is just very preliminary, and we have, what I want to show is our first efforts to be able to have electrophysiology estimation on the anatomy of the big brain. This is not a trivial task, because to be able to do that, we have to obtain good solutions for the forward problem, so the estimation of the leak field. And I am going to show two of the efforts. The first one, let me see. So this is the first one. It was created at the Cuban Neuroscience Center for Electrophysiology using the quantitative EEG analysis toolbox that is, by the way, already inserted in the C-brain. In this case, what we did was to create the big brain as a new template. This is a co-register to the MNI, to the average centroid of the MNI. And what we used in this case was to use the scalp segmentation for the big brain. And with this, we calculated the same type of leak field, volumetric leak field over the gray cortex, using the co-register segmentation of the brain to the centroid brain, and we calculated the leak field. With this, we were able to obtain. This is how it looks. And with this, we were able to obtain, for example, the estimation of the spectra, the locus spectra at the gray matter source using the big brain. And the solution we are looking at here is the for a normal subject of the Cuban human brain mapping project. So this is the estimation of the solution for the alpha peak. You can see at the top right the spectra at the electrode, at the scalp, and the solution we are showing here is at the alpha peak. In this, you can see that there is a very good solution for a normal person in this case. And this is another view. This is using the software developed at the Cuban Neuroscience Center for Electrophysiology. The second thing I want to show is more or less similar work, but done at the West, at the China lab. And in this case, what they did was to create an average, superficial leak field for the estimation of the sources of an MEG case. So the magnetoelectroencephalogram over the big brain. So for this case, in this case, they created a superficial leak field using also co-registered to the MNI template, and this is for a subject of the Human Connectome project, where we have a magnetoelectroencephalographic data. this is a comparison of the gray cortex.
Dr. Bosch, Dr. Bosch. So we are like at 4 minutes and 30 seconds already. Could you finish up, please?
Okay. Okay, this is just the solution that we got for the subject of the Human Connectome project using the BigBrain. I just want to say that this same data that we use here were used by Jorge Riera, that is going to present another work later on here. And the method for solution of this is going to be presented by Pedro Valdez. I just want to say that it's a group work, it's not just my work.
Okay, so one of the mysteries that we still don't know is how the cerebral cortex process top-down experience information and bottom-up sensory information to produce actions. And one of the most important paradigms is performance monitoring, in which you have, for example, visual information and information coming from your experience that is reflecting task rules. And then you have, based on mitigating errors, you have to perform specific action, motor action. It has been postulated by Matthew Larkin group that this comparison of top-down and bottom-up information is achieved by layer 5 parameter cell preferentially, and for that they have this phenomenon that is called sodium and calcium spike resonance, a calcium spike generated at the level of the path and a sodium spike generated in the soma. And based on the input of neurotransmitter like NMDA or GABA, this neuron can change dynamically the gain of these two distant dendritic branch. We have proposed a very simplified model of two compartments that summarize the main ionic channels needed to reproduce most of these property that this neuron has. We also have been developing a theoretical framework that connect them to the local field potential recorded with laminar electrodes. We have simulated how this current distribute across this compartment, and we have also generated many columns, thousands of these pyramidal cells, and based on that reproduce local field potential. What kind of property we already reproduce with this simplified model? We reproduce the current spike rate sensitivity of these two regions that are differentiated in the terms of the energy that have to be injected in these two regions. As seen in this figure, we also have been able to differentiate to reproduce the synaptic coincident, the input coincidence strategy that this neuron uses that is called the backfiring. And we also have been, and here you have model and experimental data next to each other, and also be able to reproduce the critical frequency that this neuron has to produce calcium in the dendritic tree due to action potential in the soma. And also we have been able to reproduce the effect that the blocking the cation occurring IH has on this neuron. Lastly, we have been able to reproduce data recorded from the Larkin group in rat, showing that this neuron produces a typical current density map that can confuse researchers, assuming that there is work by layer 2 parameter cell, while this is in fact reflecting that there is some calcium activity that have been generated in this neuron and produce this polarity reversion later. We also have been able to simulate many columns of the cortical region in monkeys. Based on the topography, we have been able to reproduce what kind of signature this neuron or this kind of activity, calcium activity spike, produce in these electrodes. So why the big brain? So we need to differentiate between this laminar input of layer 2-3 parameter cell and layer 5 parameter cell. The big brain offered a way to to differentiate. This is just a caricature here, or differentiate different cortical areas, and based on that, distribute the neurons in this lamina adequately. We have used the BigBrain, as Jorge Bosch just mentioned before, and what we did was to put a mini column in the supplementary motor area, simulating an area that is well known, participating in performance monitoring, and then we have used the co-registration that BigBrain to Collins MRI, perform the source analysis, the volume conductor characterization, and we were able to, for the first time, to reproduce the signature that this calcium spike imprint in the EEG and MEG. And I want to finish just my presentation with the video of this calcium spike signature from that particular area in EEG and MEG. And with that, I conclude my presentation.
Yeah, thank you very much. So the video is running through, I guess, or it's done. Okay, so the next speaker is Kamil Uludag from the University Health Network, Toronto.
Hello, everybody. The title of my talk is From Big Brain to fMRI, the role of hemodynamic modeling. Let's remind first what we are measuring when we do fMRI. So we have neuroactivity, spikes, synaptic activity, and via neurovascular coupling, we are measuring changes in blood flow. And via complex hemodynamics, we are obtaining changes in blood volume and deoxygenate hemoglobin. Because deoxygenate hemoglobin is paramagnetic, we are measuring this as changes of the fMRI signal called the Bolt response. Interestingly, most computational neuroscientists ignore the fact that fMRI is actually only an indirect reflection of neuroactivity. There's a model from Carl Friston in 2003 where he modeled the chain of events from neuroactivity to fMRI, and this model has been implemented in SPM and the virtual brain. Recently, we have updated the model, and the main difference between this model and our model is that we can distinguish excitatory and inhibitory neuroactivity from the fMRI signal. Now, if we go to higher resolution with CSF on top and white matter on the bottom, then we have neuronal activation distributed over the cortical depth, represented by excitatory and inhibitory neurons. Again, in the same way, neurovascular coupling changes blood flow, deoxygenate hemoglobin, and blood volume. Now, if the previous model I have shown would correspond to one layer of neuroactivity and vascular changes. However, when we go to high resolution fMRI, we have encountered an additional problem. And the problem is that the deoxygenate hemoglobin has to be drained to the surface of the brain, which leads to the fact that if we look, for example, layer 3, that if we measure Bolt activation in layer 3, it's not only correlated to the changes of neuroactivity in layer 3, but also to the changes of neuroactivity in the bottom layers, because the deoxygenate hemoglobin of the bottom layers is draining towards the surface. And this model has been published just recently. Now, the research line I'm proposing is if we take the big brain data and also additional data receptor architecture to build regional microcircuit models and also to extract vascular parameters and layer depth definitions, and using then our model to calculate for any given neuroactivity pattern, Bolt signal activation over the cortex. And if we then use experimental laminar fMRI data, use Bayesian model inversion to get back to the microcircuit and the parameters of the microcircuit. And that's it. And thank you for your attention.
Yes, thank you very much, Kamil. The next speaker is Katja Heuer and Roberto Toro from the Max Planck Institute in Leipzig and Institut Pasteur in Paris.
What is it like to be a giant anteater? I would like to present you the latest developments in our tool for visualization and annotation and analysis of histological data, MicroDraw. Histological data provides unique information on the fine structure of the brain and has been collected for more than a century for an incredible number of species. And this is just one example, the Brain Museum from the University of Michigan, which provides data for more than 50 species. There are several similar collections, like in particular the Stefan collection, which is the basis for a large part of our current knowledge of mammalian neuroanatomical diversity, which is very important for our understanding of how our brain adapts, and especially the human brain. And this is just, however, histological data is particularly challenging to visualize and to analyze and to share. And the digitalized images are very large. So the material is fragile, the images are large, and they often require lengthy manual annotation. And we've been developing MicroDraw for several years now. It's a tool that allows us to study the development of the ferret brain. And it provides a simple web interface to visualize and annotate data accessible through the web. Provides a basic project management and tools to enable researchers to create distributed teams and to work collaboratively on the same material. And that makes it so powerful, because there's so much manual work required that we couldn't do that possibly alone. And MicroDraw has a RESTful API, which allows us to query its database and programmatically enable its integration with complex data analysis workflows. And we want to illustrate that in the new functionalities we've been building in by analyzing the giant anteater histological data from the Brain Museum website. So here you can see what the website provides. It allows us to inspect different slices. However, it's very difficult to do anything more sophisticated with them in that form. And using MicroDraw, however, it's possible to turn this data into a living matter for distributed collaboration. And I will show you our current histological technological stack. And there's the MicroDraw website, which we use for the collaborative annotation and the visualization of the data. And then through the RESTful API, we can connect it to microdraw.py, which relies on several open source packages for machine learning and deep learning and geometry processing. And here, this is, for example, one slice from the giant anteater brain, and this is our annotation of the PL surface. And here you can see the result of our algorithm for the generation of inner contours and the lines we use for measuring the gray level profiles. So you see the indication of the directions where we measure. And we use the k-means to cluster these profiles. And here you can see the example of 10 different classes. MicroDraw.py is able to use automatically generate images where you can see the connection between where these clusters can be found. So here you see, for example, 1, and here in the sulcus cluster 1 and cluster 2. And so we have examples of where in the cortex they are located and their gray level here. And here you can see the surface reconstruction. So we've developed algorithms to align these different contours that we generated from MicroDraw, and then we can generate topologically correct surface reconstructions. And this is the surface reconstruction based exclusively on the data we downloaded from the Brain Museum website. And then we developed functions which allow us to project the clusters measured from the histological profiles into the surface reconstruction. And here you can see the first results where we've been able, which we have been able to obtain with MicroDraw. And we were happy to observe that our clusters were symmetric and that some clusters naturally tend to follow sulci or gyri and to distinguish kind of neocortical regions from the rest of the brain. And here is a principal component analysis of the profile similarities. And you can see clearly that our profiles allow us to distinguish the rhombencephalon and from archicortex and piriform cortex and from neocortical regions. And what's interesting,
Katja, we're at 4 minutes now.
Okay.
Can you please wrap up? Thanks.
Yes. So the most neocortical sulci look like archicortex. That was something funny we observed, and we were very happy about that. And to conclude, I could just say that MicroDraw allows us now to perform our first complete quantitative analysis of psychoarchitecture. And it's fast and lightweight, runs on that desktop or mobile devices. All you need is a website. And it's open and development at here. Like you can join us to open best practices. We are open and happy to have you joining and collaborating with you. Thank you very much.
Thank you very much. Terrific. So the next speaker is Mona Omid-Yegane and Claude Lepage from the MNI.
Hi everyone. My talk is about nonlinear registration of 1 micron histology sections into 3D 20 micron big brain space. Processing and visualization of digitized histological sections from human brain is challenging due to large image size. Big brain was originally scanned at 10 micron in plane resolution, but downsampled to yield 20 micron isotropic resolution in 3D. These sections have now been rescanned at 1 micron resolution, which allows the identification of microscopical features at the cellular level obscured at 20 micron. This allows to visualize single neurons while maintaining their topography in 3D space. We have registered 2D 1 micron histological brain sections into big brain space. The algorithm has 4 different parts. Data conversion. Scan sections are first converted from big TIFF format to MIG. A spatial transformation, a global transformation, is calculated for each 1 micron section by concatenation of 3 individual transforms. The latter 2 transformations originate from the reconstruction of the 3D Big Brain 2015 release. Blocking, cropping, and registration, where the aligned 20 micron image is divided into smaller 2D blocks, and an inverse of the global transformation is applied to the corner points of each output block to estimate the extents for cropping the block of 1 micron unregistered section. And then the global transformation is applied to the cropped image. And finally, the reconstruction step, where the registered resampled blocks are concatenated to form each registered 1 micron section. Here you see an example of blocking of a 20 micron section in aligned 3D space and corresponding blocks on the 1 micron input section not registered. The global transformation is applied on the blocks of the 1 micron section to produce the output section at 1 micron in 3D aligned big brain space. And here you see 2 examples of blocks at 1 micron overlaid on corresponding 20 micron section in big brain aligned space. The first example is for the cerebellum area, and the next one is for the cortex area. As you see, you can see the neurons in the 1 micron sections. As conclusion, I can say that cutting an image into blocks allows resampling of full histological sections at 1 micron. Previous tools could not operate on full sections due to prohibitive memory demands. By blocking 10 by 10 each slice, we will have almost 8 minutes to transform, which uses almost 1.5 gigabyte memory, which is affordable. And also the block concatenation took almost 1 hour. The total processing time per section is about 15 hours on a single core without any memory bottleneck, allowing simultaneous processing of multiple sections on multiple core compute nodes. Mapping 1 micron to 2D histological sections to the 3D big brain will offer new opportunities to locate and characterize individual neurons, as well as map their distribution across cortical layers, areas, and subcortical nuclei. Thank you.
Many thanks. That was terrific. So the next speaker is Valerie Hayot-Sasson from Concordia University.
All right, so hi, I'm Valerie. I'm going to talk to you more about improving the processing time of neuroimaging workflows. So my specialization, or the project that I'm currently working on, is building a hierarchical file system for efficient pipeline processing. I hope that this will be, or that you'll find this useful to you as with the increase in size of BigBrain and the fact that your workflows are becoming more and more data intensive, I hope that this will improve the processing time. So our workflows are more data intensive, and what happens is usually what we end up using is high performance computing resources, or we go through a portal like CBrain, which connects you to a high performance computing resource. And these resources are, these clusters typically leverage a shared parallel file system, which means that when you want to interact with your data, you have to, when you're on your compute node, you have to make a request to a server, a storage server. So you have to go across the network and then be impacted by the fact that your data would be written to hard disk, let's say. So there is a cost to interacting with your data on a shared file system. And also the shared file system may be, such as Lustre, may be shared by many different users. That also impacts the cost of interacting with such data. Now, big data file systems, what they've done is they've tried to reduce the IO cost by not using a shared parallel file system. So what they do is they use data locality. That means that when you process your data is located on the compute node where you're processing your data, and they also try to maintain it in memory. So instead of using a SSD or an HDD, they'll have your data in RAM, which will give you optimal performance. So here I have one of my previous works where I showed, so basically in essence, I was copying BigBrain, the 40 micron one, 10 and 100 times over. And especially at 100 times over, what you see is that there's almost a 4-time difference between processing in memory and using a shared file system such as Lustre. So what my project does, or what C is it's a hierarchical file system that tries to give you that in-memory computing performance similar to big data engines such as Spark without making you alter your workflow in any way. So this is kind of what it looks like. We have this lightweight library that intercepts libc calls. So libc calls are like calls that you make to open a file, whether it's in Python or whatever. If you're on a Linux system, you'll interact with libc. And what my code does is it intercepts these calls and then switches the directory or finds the best directory for you to write your data or interact with your data. So you have this view known as a mount point. That is where you read and write your data, but no data is actually written or read from the mount point. It's written from other file systems such as memory, SSD, HDD, depending on what's the most efficient that's available. And you specify the file systems that you have available, such as memory, SSD, HDD. And also, if you'd like to have that data materialized to Lustre, you can also specify that. So this is kind of what it looks. You have your mount point, and it gives you a view of all the files within C. But these files can be distributed across many file systems, such as TempFS and SSD. And what you see is, so when you interact with the mount point, you'll see all your files, but you won't necessarily know where they are. So when you say open within the mount, you can open from TempFS, or you can be opening from SSD. And so I've done a bit of testing. It's still very much a work in progress. So what I did was I had one BigBrain file that was of 617 megabytes in size, and I had 20 iterations, so just copying over and over 20 times.
Valerie, we are at 4 minutes and 20 seconds. Could you wrap up, please? Okay.
Yes. So, and then, yeah, so what we did was just copied it over and over again. And what we can see is that it's almost twice as fast, C is almost twice as fast as Lustre. And it's very comparable to writing directly in memory, which means that there's not much overhead to the library. And that's it. My code is available on GitHub. And if you believe that this could be interesting to you, please contact me. Thank you.
Thank you very much. The next speaker is Sherri Barakat, and he works at Jülich and University of Iceland.
I'm Sherri Barakat. I'm a PhD candidate from the University of Iceland and from Forschungszentrum Jülich. And I'm part of the team that's working on application co-design of a modular computing architecture for cellular big brain, connecting the Canadian sea brain and German supercomputing infrastructures. So our work is centered on setting up modular computing architecture for cellular big brain. And the first step of our work is to set up the workflow based on which the communication is going to take place between the available infrastructures at Jülich and at Seabrain. So after that, we optimize for access to this data that's going to be stored on the servers. But for the initial work, we're working on a draft architecture for the joint infrastructure between JSC, which is the Jülich Supercomputing Center, and Seabrain. It's expected to be done around now, and we'll discuss this in the next slide. So the current draft architecture for the joint infrastructure is visible in this chart. At the bottom, we can see the computing resources with the pre-installed software from both parties. They each have their own schedulers and the required tools for running the code. And access to these resources is via their own individual authentication protocols. But Seabrain uses group accounts and Jülich uses group accounts and federated identity. The communication between these architectures is going to be done at the level of the data access, which can be seen here. Is going to be done via SCP and grid FTP. And at the same time, we're going to implement some type of version control system for the software that's going to query the database. Afterwards, the data access pipelines are in place, and they take charge of relaying the data to the clients, either via SSH or through the Seabrain portal. The tools that we're going to use for all of these processes are presented here. We're going to use SnakeMake which is a workflow management system for scalable data analysis. DataLad, which is a versioning system built on Git Annex, and it integrates also into Python. And we're also going to use Boutiques, which is a cross-platform tool for automatic publishing, integration, and execution of command line applications. So after all of this is done, the next steps is to refine the draft architecture and then start working on data access protocols for all parties involved. And with that, I'm done. Thank you for your attention.
Thank you very much. Terrific. The next speaker is Christian Schiffer from Forschungszentrum Jülich.
Hi, I'm Christian Schiffer. I'm doing my PhD at the INM1 at Research Center Jülich, and I'm developing algorithms for automated mapping of cytoarchitectonic areas in histological human brain sections. Timo Dickscheid already talked about this and gave an overview over the methods we developed earlier today. And in this talk, I want to give you an overview over one of the methods that we developed and are currently using. As a reminder, we are trying to train artificial neural networks to identify cytoarchitectonic areas, here given in color, given high-resolution histological sections. We usually here use the 1 micron scans of the histological sections. In a perfect world, we would like such a model that we trained to be able to segment arbitrary cytoarchitectonic areas across several brains. But it turns out that it's actually quite difficult. And this is partly because we have a high intersubject variability across brains and also differences arising from different processing protocols. So our idea was to pursue a divide and conquer approach, where we first subdivide this very complex task into smaller tasks which are easier to solve. As a first step, we focus on just one brain. This brain is composed of several thousand histological sections. But even when only considering one brain, we still have a high intra-subject variability. So as a next subdivision step, we focus on just a small substack of sections. Such a stack may, for example, be composed of, in this case, 120 sections. And then we assume that only the outer two sections of the stack are annotated by a neuroanatomist. We then use these two annotated sections to train a convolutional neural network. And by training only on these two sections, the network highly specializes on the local cytoarchitecture and morphology of the sections. And in the best case, can then be applied to all sections in between. So in this case, this would mean we would need to annotate two sections and would be able to map 119 sections automatically. When we can solve this small task, we can go up again and train one specialized CNN model for each local substack of sections. We already applied this method to 18 areas in the big brain and also to other areas in two different brains, one also silver stained like the big brain and one nickel stained, demonstrating that this method can also be applied to brains which were processed with different processing protocols. In particular, for the big brain, we were able to reconstruct 4 areas from the visual system in 3D at 20 micron isotropic resolution. And in the future, we plan to use this method to reconstruct even more areas. And with that, I'm finished. Thank you very much.
Thank you very much. That was excellent. The next speaker is Andrea Brandstetter, also from Forschungszentrum Jülich.
Hello, everyone. My name is Andrea Brandstetter, and I will present you how we mapped and visualized a small subcortical structure supported by a deep learning model. The structure was the lateral geniculate body. It is part of the visual pathway, and you can see here on the image on the top that it can be subdivided into several layers. To give you a short overview about what we did, so we took high resolution flatbed scans. We annotated every 30th section of every layer of our structure using microdraw. And we trained the network that was just presented by Christian Schiffer. So it was trained to recognize the cytoarchitectonic pattern according to our manual annotations. The model then predicted delineation of each layer on every section of our structure, and transformations then were calculated and applied. So we also had these predictions in 3D. Finally, we visualized our results using ParaView. Taking a closer look on the lateral geniculate body, you can see that it consists of 6 distinct layers, of which layer 1 and 2 are contain magnocellular neurons, and layers 3 to 6 are considered as being parvocellular layers. On the right-hand side, you can see now the results, so all the 6 layers in 3D. And you can also see, of course, the overall shape of the whole lateral geniculate body. And also to give you some numbers, in total, we had almost 1,500 annotations on 365 sections. As a conclusion, so this was our first application of deep learning for a small subcortical structure. The deep learning model is not only faster than we as we would be doing everything manually, but it also predicts the annotations on each single sections, whereas an interpolation would merge existing manual annotations only. So especially for heterogeneous and small structures, this deep learning approach might be more accurate, but to be honest, we don't know this yet and not for sure. And therefore, we are very excited to compare these results maybe with Atelier 3D. But of course, we would also like to try this deep learning model for other subcortical structures. I would like to thank to Najwa Bolakreef for the mappings and also Christian Schiffer for the collaboration and you for your attention.
Thank you very much, Andrea. So the next speaker is Kai Kiewitz, also from Forschungszentrum Jülich.
My name is Kai Kiewitz from the Cecilia and Oscar Vogt Institute of Brain Research in Düsseldorf, and I'm really happy to have the opportunity to share with you some insights in deep learning networks that reflect cytoarchitectonic features that we use in brain mapping. As you've heard multiple times today, especially in the talk that Timo Dickscheid gave, is that the current gold standard cytoarchitectonic mapping techniques that we use are not feasible anymore for big data sets like the BigBrain data set, for example, that a lot of us are working on. So one possible solution to overcome this has been introduced by Christian Schiffer, for example, a few minutes ago. That is a deep learning based tool for cortical area segmentations. And what we were interested in when we applied this this interesting tool is, how does this tool actually operate? So what we wanted to see is there a resemblance of learned filters inside the tool that actually resembles cytoarchitectonic features? So we took a look into the tool, and what we saw is that on superficial layers of the tool, which has a convolutional neural network architecture, we found filter activations for cells, which you can see on the right side. We also found opposite filter activations for example, the cortical layer 1 and the white matter, which you can see in the middle. So already a first sign that the tool incorporates some cytoarchitectonic features. If we go deeper into the tool, we find filter activations for cortical layers. This is an excerpt from the primary visual cortex, and what you can see on the left side is a filter activation for cell-sparse cortical layer, and the composing filter on the right side shows you a filter activation for the more cell-dense cortical layers. And if we compare these filters to the GLI approach, which is the current gold standard technique for cytoarchitectonic mapping, we see that it actually fits quite well to the profile shapes of the GLI. Running towards the final layers of such a CNN tool, we find filter activations for whole cortical areas. This tool, this network was trained for a cortical area HIBALL. So in the later layers, we find filter activations for the primary visual cortex, for example. We also find some opposing filters, which you can see right here. This filter shows activation for the cortex around HIBALL, which is this part of the cortex right here. This filter right here shows an activation for the cortical area HIBALL, which is the primary visual cortex, but no activation for the surrounding cortex. And I'm sorry, if you combine these two filters, you see that there might be a border representation inside the trained neural networks that actually quite well fit to the delineations based on the current GLI approach. So we think that filter activations of a deep learning approach indicate that there is a resemblance between learned filters and traditional cytoarchitectonic features. And these filter activations also compare well to the currently used GLI approach. And this is why we think that these findings validate deep learning-based biomapping as a semi-automatic alternative for high throughput mapping workflows in the future. And yeah, this is it. Thank you very much.